Hello, everyone. Welcome to the December monthly meeting. I'm excited to introduce Dr. Ravi with us, a distinguished expert in genomics and bioinformatics. He's currently serving as an assistant professor, formerly trained as a physicist. Dr. Ravi has seemingly combined physics and genomics to address some of the most intricate challenges in genetic research. His groundbreaking work spans mitochondrial genetics, the dynamics of the T cell receptor, and the role of piRNAs in transposon control with the germline. Dr. Ravi has also pioneered innovative technologies, including unbiased small RNA sequencing and advanced methods for sequencing mitochondrial DNA and TCR repertoires, which he will be going into more information later on. And with a rich history of impactful contributions, including research on mRNA splicing and the development of widely used bioinformatics tools like SpliceRag and Kismet, Dr. Ravi has made significant advances in our understanding of genomics, and we are honored to have him here with us today.
Thank you, Salah. So I'm going to quickly go over a few what I call vignettes. And obviously, I cannot talk about everything, and I don't want to just sprinkle things. So I'm going to go into detail in one story, and let's see how that goes. So, okay, I started out in physics. Actually, I started in engineering as an undergrad, and I learned. So I'm just showing you things I learned, like electronics, Boolean logic, and things like this. In physics, I looked at phase transitions and buckyballs, and then I switched to genomics. And now you may ask, okay, you did all these other things, and what's the connection or why? And uh, partially, I want to show you how these prior training helped me solve some problems in genomics. But secondly, I think I mostly like to find questions and answer them. That's my style. I some challenges, and there are lots of challenges in genomics that I could address, and that's why I started doing genomics. So in the early days, I got in when genome sequencing was just starting out in some sense. Not the sequencing itself, but like the large scale sequencing, where you sequence the whole genomes of people, of animals. And I helped build the first human SNP. SNPs are single nucleotide polymorphisms. That's the differences between me and you, for example. Like if you compare the DNA, you'll see differences. And that I compiled one of the first compilations of this for humans. And then I built a library for silencing. I did splicing. As you mentioned, I built many tools. So, but the key point here is that I was working with people and helping them and doing things, but it was not like I was driving the questions at this point. I was but also I didn't know much about genomics. I was learning things. I was helping where I could. I did make some sort of advances in thinking about splicing signals, but mostly it was building tools. And the next phase is where I started, let's call it doing science. And the way we ended up doing science was I established a lab that started where I started doing actually generation of data myself. So we actually started sequencing things. And the cool thing here is that we got in with like literally opening manuals and reading. Uh, like we are amateurs at this point. Then I knew about data. And one of the first things we did was to sequence the small RNA because I knew that data sets. I had worked on piRNAs. I knew a lot about it. But it turns out that the method we use, and I'll go into details now later, is were all biased. So it would like pick one more than the other, and so, but no one knew this in the field. And when we first discovered it, we were actually very scared to like even say that we found that because big experts in the field are all working. No one has seen it, and we're the first people to see this kind of bias. So I first thought I was wrong. And me and the student, we spent like 6 months thinking about the data, and then eventually we concluded that it's correct, and we published it, and that actually resulted in a patent. And actually, that's the method that's used for sequencing small RNAs nowadays. And after doing that, we got some confidence in doing this, and that's how we ended up thinking about other questions like mitochondrial DNA, and we invented a method of sequencing the mitochondrial DNA accurately, and that led to actually a discovery of that if you look at different cells, they actually exchange mitochondria between them. And unfortunately, today I won't have time to go into it, but we can talk about that if you have questions. And right now, we're actually working on this using the mitochondrial DNA as a marker for cancer, for just looking at what happens when you get something like Parkinson's. Yeah, what's the effect of mitochondrial DNA changes on these things? So then we moved on to looking at the TCR repertoire. And the reason we're looking at the T cells are in your blood, they are actually the first line of defense. So when you get an infection or something, the T cells are the ones that identify the problem and attack the cell. So when you get COVID, the cells are producing COVID proteins, which the T cells end up recognizing, They start killing those cells that are infected, and they help B cells produce antibodies. So if you don't have T cells, you'll die because T cells, without T cells, but you can live actually without B cells. And there are a lot of people, older people, who may not even have B cells producing antibodies, and they need to get injections of antibodies and things like this from other people. But you can survive without B cells, but you cannot survive without T cells. And we developed a method of profiling the repertoire. Repertoire is the full collection of T cells. So each T cell, this is actually very interesting. So this is interesting to your audience, I think. So if you take a T cell, this is one of the few cells, along with B cells, that don't have the genome that you're born with. So they actually rearrange their DNA. Each T cell has a rearrangement in the thing, and that's what allows it to recognize literally infinite kinds of invaders. So that, I mean, this was a big mystery in the beginning. Like, the genome is all static. How the hell is your immune system, your adaptive immune system, how is it able to recognize almost anything that you can throw at it? And the secret is this rearrangement of the DNA that happens at each cell. So your T cells are very different from what you were born with. Not very, but that one receptor is different. And so we are sequencing and profiling this collection of receptors, and that's called the repertoire. So the repertoire is just a collection of T cells in your body, and we are profiling it to see what can we gain from profiling it. And for example, we can look at the response to vaccines. So we showed that we can very quickly, like within a week of getting the COVID vaccine, you can actually look at the T cell repertoire and see if there is a response, and also predict if the person is going to robustly produce antibodies against the COVID uh, virus. We've done work on humans, dogs. We've shown, for example, that dogs have breed-specific signatures. We're thinking about autoimmune conditions, and could we use this to monitor that? Another area we worked on was looking at Ebola virus. So I'm sure you guys have heard of this. It's If you get infected with Ebola or Marburg, you're going to die for sure. It's a very dangerous virus, and there were like about 4 years ago, there was like a big scare. People got
Yes.
infected, and so one of the interesting thing about this is bats can get infected by these viruses and nothing happens to them.
Wow.
They will literally shake it off.
Yeah.
Whereas humans are like immediately dying.
Mhm.
Why? So we started asking this question, what's the difference? And actually this led us into like a whole trying to understand how does your immune system react to viruses? What's its connection to blood pressure? Uh, there are things called macrophages in your liver, all these in the iron metabolism, all these are connected. So in ways that are very interesting. So one of the theories that is kind of now gaining ground is that maybe malnutrition in poor countries leads to iron deficiencies, and that may potentially be why people are more susceptible to infections because you don't have enough iron in your system.
Yeah.
Um, yeah, so there are things like this that you can investigate. So these kind of problems led us to these kinds of interesting questions.
Yeah. Sorry, I want to interrupt you on that note. I would
Yeah,
have a question basically. It's like kind of like a personal question for you.
yeah.
Which out of all these topics did you find most interesting? And also, do you find like in the future there could be a lot of more work done in future studies?
So each of them is interesting. I mean, that's why I'm showing it to you. Like we've
Yeah,
done
for
lots of
sure.
other things.
Yeah,
So I pick these things because I find them fascinating and
sure. Yeah.
interesting and challenging.
Mhm.
Each of them can go. So unfortunately, in this day and age, all of this is driven by literally money.
Yeah.
So can you get funding? Can you get almost it doesn't matter if I'm interested.
Yeah. Yeah.
Can I get someone else interested in my question enough to say, okay, I'll pay you, I'll give you some money, or I'll invest in you, or I'll support you through the government grant. So this is a big problem in science in general, that everyone is struggling to convince someone else that what they're doing is the best thing on earth and you should give them the money. So I would claim, oh, if you gave me all the money, I would solve this and solve that. But there are 100 other people doing the same claims. So this is actually a big problem. But yeah, so I think, for example, like the mitochondrial DNA, we did that work a while back, and then now I'm coming back to it because I was able to get clinical samples. And that allows me to now start thinking, how can I apply it in the clinic? Can I use it to help patients? The TCR work, again, we have done a lot of work on dogs, humans. I have lots of ideas. So if I can get funding, I would start working on that. That's something I really like, that whole topic, the T cells and how they interact with vaccines and
Yeah.
with infections.
Sure.
Um, Yeah, so almost what am I showing you? I would love to pursue more if I
Perfect.
had the if I had funding for it.
Mhm.
Um, so I want to now go back to this one, and I want to show how my training kind of helped in solving a problem. And I mean, this is partially to say that what you learn now may be useful. Someone will say, why should I study geometry or what's the point of this or that? But you, all knowledge is good. Something, it'll eventually come out somewhere and you can use it. And that's why it's fun to learn things no matter what, even if it's not directly applicable. So I just want to show you one project that we worked on. This is the earliest one, and I'm kind of proud of this because we entered the field when we didn't know anything about sequencing in the lab, and we set up a lab and started sequencing things. And so I'll just explain the problem. So when these new machines came out, so the big change in sequencing was you could sequence one DNA molecule at a time.
Mhm.
Now you can sequence millions of them at a time, and that's the big difference. So when these machines came out, but initial machines were limited to 30 base pairs. That means 30 nucleotides. You could just read 30 letters in your DNA.
Okay.
But that's good enough for small RNAs. And they didn't have things like, so the capacity of what is called a flow cell is 1 unit of run, let's say. You could get like a million reads. So that doesn't give you much room, but you can still, uh, for things that are not very complex, you don't want to waste the whole thing on one sample. So you want to add many samples. But to do that, you need some way of identifying each sample. So what we would do is what are called barcodes. We attach to the molecules things that are identifying each sample. So I take your sample and put a barcode like ACGTT, and then I'll take my sample and put another barcode. Now, when I mix them together, I can sequence them and then use the barcodes to separate them out again. And this allows me to then, it's called multiplexing, and you can sequence many samples at one shot. So we decided, okay, small RNAs, we don't need that many reads, so let's multiplex them. So here's an example where we took the same sample, split it into 5 parts, and then we gave the same different barcodes to each one, okay? And we ran it. This actually happened by accident, but I'm just showing you the experiment we repeated to show that this is consistent. So initially what we did was someone gave us samples and 3 of them were the same. We put different barcodes, we threw it in, sequencing comes back, and the results are wildly different. And of course, the person who gave me the samples are like, you guys don't know what you're doing. This is garbage. It has to be exactly the same. Why? And then we got very worried. Okay, what are we doing wrong? So we went back and repeated the experiment. We took a sample, split it into 5 parts, gave each one a different barcode, ran it. And what came out was that if you look at this here, so one means the two are exactly the same. They're correlated. They're the same level of expression. So you can see that the same samples with different barcodes have different levels of correlation. Zero point 0.6 means it's similar, but not 100%, like 60% similarity, let's say. So this was a mystifying thing. Why the hell is it different, even though we know we put the same things in? And we know it's not sequencing platform because they're all sequenced on the same machine at the same time. So it must be in the sample prep somehow. So that was our conclusion. So we said, okay, let's investigate. So this is I'm just showing you a quick picture, but the key idea is you take a small molecule, you attach a barcode adapter on one side, and then you attach an adapter on the other side. And now you have two anchors that you can use to amplify, and that's what is used for sequencing. That's what the machine uses to read. So this is the protocol, and this is used by all labs in the world. So we did that. And actually, when we started looking at the literature, people are all like, "Oh, it's very mystifying." And then we realized many people had noticed a problem, but no one had actually dug into it. In fact, there was a paper in Nature where they claimed that different methods of profiling lead to different results. And unfortunately, depending on your problem, you pick the method. And this is very unsatisfying to me. Like, how the hell can it be that a method matters of how you measure things? So it's like, okay, we have to figure this out. So um, yeah, so we started looking at different steps in this process and said, where can it come in? And there's a ligation of adapter on one side, ligation on the other side. Where is it entering in the system? So we first thing we did was there are lots of PCR. PCR is, I hope everyone knows, it's a way to amplify the DNA. And we used PCR in this process. So we said, okay, is it because of PCR? So we did PCR for 18 cycles. Each time you do a PCR each round, you get doubling. So PCR 18 versus 25 means you have 7 rounds extra in one case. But we found that the correlation was very good. There was no difference depending on how much you amplify. So this says that it is not coming from PCR. Okay, so then we figured out that both ligations cause bias, and then we actually explicitly showed that depending on what you ligate, you get a bias. This is very lucky actually, but today you won't discover this. The reason we discovered this was the barcode was right next to the where we were ligating. So it was able to see the barcode. Nowadays, the barcode is moved inside, and so you're ligating with the same end of the adapter and the sample. So then you won't notice this. So it is a kind of an accident of technology also that we were able to find this bias. And then we showed that if you took multiple adapters with different barcodes and put them in a pool, your bias disappeared. So then we came up with this idea that you should synthesize the adapters with ends in the end, which means you randomly have a mix of things at the end. This eliminates the bias, and this is actually the accepted method of sequencing small arrays now. So we were kind of proud of it because we came with no knowledge and we were able to make an impact on the field. And now there's a company that actually sells kit based on this work. So it's kind of satisfying that the very first project we started, we ended up with something very interesting and impactful for 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 the field and for so this has implications for clinical analysis. If you want to go and look at small RNAs in a blood sample, you would use our method to characterize the sample. Uh, yeah, and it's interesting because now then we went back and looked at all the evidence that was there in the gel shifts and things like this. We noticed that you could have seen this if you had paid attention, you'd have noticed there's a problem long before we found it. But the thing is, most people didn't pay attention to the various signals that are there in all the various experiments that are done. So sometimes, so here the idea of randomizing the end, that all comes from, I think because I had a physics training, I could think of these ideas. Um, so so this is, I just hope it gives you a flavor for how, uh, you can come into a field being an amateur and you can end up making an impact. So I would encourage all of you to think from first principles and actually dig into something crazy you notice, I don't know, and you start digging in and
Yeah.
you end up actually discovering things.
So like on that note, basically, what basically advice would you give to someone that found your research interested, and they're interested in genomics? Where do you think they should start? Obviously you started from a physician standpoint. So for them, where would you, like
Yeah,
suggest?
so find what your strengths are. Like, so for example, maybe it's analysis. Then there's lots of data sets. You can start playing with the data sets and thinking about questions that uh, definitely read. Um, Nowadays, you have lots of sources like Twitter,
Yeah.
blogs. So keep an eye out, follow people who are doing research and are talking about things. Reading papers is a little bit too much because there's just so many papers coming out and they usually are at a level that's too high. Even I don't read papers because I find them always too much is assumed going in, like you know the field. So, but if you read discussions, things, you have an entry point. You
Mhm.
can like get into things and start look at people's code. So if you're going from that end, you should start looking at code. You should start trying to write programs. And now with laptops, you can look. I do most of my work on my laptop. You can actually do a lot of analysis by yourself. You don't
Yeah.
need anyone's help.
Perfect. All right. Thank you.
But and then the other side is if you want to do from the lab side of things, let's assume your talents are in the lab side of things. So then I would say go and do some kind of internship somewhere. Go work in a lab. Make sure that you're going to a lab where you feel their interests are somewhat aligned with you.
Yeah.
Because often as you're entering, look, no one is going to give you the most challenging job. You'll have like a technician's work, or like you'll be doing very menial tasks. So make this and do that, and slowly you'd absorb and then move on or move ahead. But you can't jump into or parachute into the you can maybe, but it's more difficult. No one's going to say, "Hey, come here and let's do my most critical experiment I want you to do." So you need to build yourself up in some way, both sides, both computationally and in the lab.
Mhm.
So start with internships somewhere if possible.
All right. Is that it for the slides, or is there more?
I had lots of slides, but I just want to, yeah. So main thing I think the main thing I wanted to point out is that because I change, I look at new things all the time. So you were always, at least I feel I'm an amateur.
Mhm.
And then that feeling I like. Some people follow one, so that's what there are many styles. Some people will take one thing and spend all the time doing only one thing or one problem and go deeper and deeper and deeper. I like to be looking at different things and seeing different problems and see where I can contribute or what I find interesting. So that means necessarily you don't have all the background always because you're learning always. And so, but in a sense, it's useful because then I'm willing to learn. I ask questions, I expose myself. I don't feel embarrassed about saying, okay, I don't know this subject because yeah, I really don't know. So I'm not expected to know. I go there and ask questions and learn from people. So sometimes it's good to have that feeling, I think, of wanting to being an amateur and learning things. And I think that's something you should have all the time. Then it makes things fun.
Yeah, sure.
Yeah, that's about it. So I don't think I have much more.
All right.
I can talk about other topics, but yeah, I think that gives you a good background to start thinking about what questions you would like to
All right.
answer.
Okay, so let's go back to some questions from the chat previously. So, basically a broad question that's, a lot of people have targeted is basically what inspired you to find this field, coming from a physician's standpoint?
Yeah, so sorry, you used the word physicist, which is different from physician
Yeah.
because physician is a doctor. I'm like
No,
more
physician.
a
Yeah, sorry.
yeah. So uh, again, it's random luck in some ways because I had a very naive. Okay, so that's a big problem. It's because I knew nothing.
Mhm.
I thought, oh, okay, I can use some ideas on physics. I can use some computers, blah blah blah. Look at GNA, genome. Uh, I had very naive ideas, and I wrote to some labs and said, "Hey, can I come work here?" And one lab said, "Yeah, come work here." And that's how I ended up. So it was purely me willing to go and look at different things, writing to people and saying, "Hey, I know nothing, but I'm willing to come and learn. I would like to do this." And someone taking a chance on me. So that's very important.
Mhm.
Because the tough part is breaking into anything is that people won't give you a chance. So you need to somehow find one place of entry somewhere.
Mhm.
Um, so then once I got in, and it took me a year or two to learn. So it's not trivial to learn enough to be able to even do interesting things. Um, so I think that's the problem that you can't know, right? It may look interesting from outside, but there's so many dirty details in going from something that's where you are and where it is interesting.
Yeah.
And you have to be willing to spend the time and effort learning, I think.
Mhm.
There's no shortcut. Like you can't just immediately say, okay, I'm going to go and do the most interesting thing here.
Sure.
And that's a problem because everyone goes to the idea, oh, science must be exciting or this must be exciting. And then you go in and you're like, oh, very detailed
Or-
things. Things don't work and you're debugging some problem and it takes time. So you need to have patience to go
Mhm.
through that.
Perfect. Okay, and one last question that was, majorly focused on, just purely for people that are interested in genomics. So basically, what current genomics topics do you think have like the biggest future and potential research in that you find? And again,
Well,
this,
like, I mean, okay, I don't want to speak for the whole field,
yeah,
but
this would
what,
be,
like, for example, what I see is like the repertoire, trying to understand. For example, okay, here's a kind of a science fiction question.
Mhm.
I could take your blood, and maybe in the future I could tell quickly, oh, you're being infected by COVID before it even shows a symptom. Just by looking at your T cells, and maybe I can tell you, hey, there's some, or
Yeah,
even before the tumor becomes big, I can look at your blood and say, I think there's something going on. Let's check. See a tumor, signatures of a tumor. That's science fiction, but that would be a goal. Like, can we do this?
yeah,
But there's lots of questions in there in terms of can you use machine learning to do this? What kind of data do you need? So there's so many arenas in that big question. Or how do you, for example, now they're trying to sequence everyone who goes, or the day you're born, you get sequenced, let's assume. Now there's a big data problem. How do you handle all this data? How do you efficiently do this? Do you need to keep all the data? What kind of data should we get? So there are questions like this. I mean, everyone is
yeah,
working on them. It's not, but there are, can you efficiently use all the computing resources in a distributed manner so you don't, I can sit on my laptop, type some things, and then it goes and tuck, 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 collects things. So there are things like this that I think that are open. There are, of course, many algorithmic small questions, but those are details, I think. But overall, the big thing is large data sets and more intelligent use of the data sets that you're generating.
All right. Well, thank you so much for your time. Thank you guys, everyone, for whoever is watching this, and thank you. This was today's December meeting, and thank you, Dr. Ravi.
Thank you. Bye.